Right, a very good morning and warm welcome to all of you. And on behalf of Department of Pharmacology, Chalapati Institute of Pharmaceutical Sciences, we cordially welcome you all to this international webinar on recent advances in preclinical research. So we know the preclinical research plays a major role nowadays because everything has to be evidenced. So each and everything, in order to launch a new product, in order to launch a new medicine, we want a strong evidences as a base. So regarding this, now a pandemic is also there, COVID-19. All the different countries are urging for a vaccine. So based on this, we have a evidence, a strong evidence has to be submitted and has to be there. And this preclinical research and preclinical area is supporting a lot for these type of evidences. And on have this knowledge in order to support these all things, we on the Department of Pharmacology, Chalapati Institute of Pharmaceutical Sciences, Andhra Pradesh, we are having this international webinar on the recent advances in preclinical research on 6th June 2020 from the 10 a.m. to 12:40 Indian Standard Time. And we have four speakers today. One is Dr. Aditya Garu, as well as the second is Dr. Shenishwarani and Andy Garu, as well as the third one is Dr. Kalirajan, as well as the fourth one is Dr. Vijay Pandey Garu. Two of the speakers are from Malaysia and they are in live, and the other two speakers from India. So well-known speakers for the preclinical area as well as preclinical research. So let us have the session. So before starting the session, let me invite our head of the Department of Pharmacology, Dr. Narendra Babu Garu, to invite the first speaker, Dr. Aditya Arya, and also to start the first session. I request Dr. Aditya Arya to start the session. Before this, I request our HOD to welcome by reading his profile. Thank you. Sir Narendra, sir, you can unmute yourself and you can come to live. Narendra, sir. Okay, that's no problem. Let me have the profile. Let me have the profile of Dr. Aditya Arya Garu. Aditya Arya is an associate professor of pharmacology and therapeutics at School of Medicine, Faculty of Health and Medical Sciences, Taylor's University, Malaysia. He also holds an honorary academic visiting associate professor position to the Department of Pharmacology and Therapeutics, the University of Melbourne, Australia, and Liverpool John Morris University, Liverpool, UK. Previously, Dr. Aditya was employed as a senior lecturer of pharmacology of Faculty of Medicine, University of Malaysia from October 2010 to January 2016. Education and affiliation of Dr. Aditya Arya is a PhD in pharmacology from University of Malaysia on cellular and molecular aspects of anti-diabetic candidate. His research work is identified by the Ministry of Science, Technology and Innovation, Malaysia, and awarded fellowship in October 2015 to join Malaysian Institute of Pharmaceuticals and Nutraceuticals under National Institutes of Biotechnology as a research fellow to establish cardiometabolic research laboratory of global standard using cutting-edge technologies. And he acquired many international awards. Recently been awarded with the prestigious civilian award, Hind Ratan Award by Government of India in January 2019 recently. He is an active member of numerous committees, advisory boards, and serving as reviewers to various prestigious peer-reviewed journals of repute. He has published 60 over high reviews, high impact research papers, and has many leadership responsibilities, including current roles as a group leader of quality of life as well as co-investigator to obesity and well-being program. Moreover, he has had acquired several national level research grants such as Fundamental Research Grant Scheme FRGS, High Impact Research Grant HIR from the Ministry of Higher Education MOHE with more than 14 million RM. Research focus, cellular and molecular pharmacology or biology using cutting-edge techniques and multifaceted in vitro and in vivo approaches. His group is critically involved in identifying specific mechanisms that underlie the development of diabetes linked with cardiovascular dysfunction. His team target insulin networks and the role of signaling pathways involved in pancreatic and metabolic functions by exploring the transitional relevance of molecular biomarkers in the discovery of potential therapeutics for diabetes and cardiometabolic disorders. So this is a short preview of our first speaker, Dr. Aditya Arya. Let us invite Dr. Aditya Arya to start his first session. Thank you so much.
Thank you very much, Mr. Tony, for your kind introduction. And I would like to thank Department of Pharmacology, Chalapati University, Dr. Pandi, for inviting me to talk about my research expertise at this forum. So please allow me to start my talk now.
Yes, sir.
Okay, should I start now? Hello?
You can start now, sir. Proceed, sir. Thank you.
Okay. Okay. So today I'm going to talk about potential therapeutics targets that are involved in diabetic research. So as we are aware, we are facing a big pandemic now, that is COVID-19. But as you keep away our pandemic COVID-19, we are also facing difficult mode of cardiometabolic condition that has been developed due to diabetes. So diabetes is also one of the epidemic condition in which many people around the globe are affected due to diabetic condition, due to metabolic process, due to some of the sedentary lifestyle. We are facing these issues in terms of when we talk about all the yes. So what happens is that there is a huge consequences developed due to this cardiometabolic stages. So as we are aware, in the world, there are around 422 million adults who have diabetes. Diabetes may be type 2 diabetic condition, type 1 diabetic condition, and so on. So we have to bear in mind of the possibilities that could lead to further consequences because of this condition. The important is type 1 diabetes, type 2 diabetes, and gestational diabetes. In type 1 diabetes, as we are aware, it is due to the massive destruction of pancreatic beta cells. So there is no much release or secretion of insulin from the pancreas. However, in type 2 diabetes, there is insulin secretion from pancreatic beta cells, but this insulin is not properly utilized by our body, so which lead to type 2 diabetic condition. Moreover, in gestational diabetes, this is a temporary stages developed during pregnancy in women. So in most of the cases, this condition is considered to be away when woman delivers a baby. So this condition, however, in some conditions and some stages, this develops further as well. So this, to take into consideration, we must think of finding a specific and profound target so that we can come across with therapeutic agent that could lead to the management of these diabetic type 1 type 2 diabetic conditions. So if you see the global, in the global context, the statistics itself explore the possibilities and narrate that there is a huge potential in diabetic research that could be placed within the industry as well as in academia to combat certain issues. As we see, not only our Asian population, it is spread as an epidemic throughout the globe. So the specific data are also illustrated here, but there is a urgent need that has to be placed in order to come across with or to develop certain medication out of that. As having said, in diabetes is a condition where in type 1 patient, there is exactly failure of pancreas that produces insulin. The beta cells which are produced, which produces insulin, are mostly destroyed, are destructed. So there is no response to the insulin secretion. However, in type 2 diabetic condition, there is a secretion or partial release of insulin from pancreatic beta cells, but that insulin is not properly utilized or converted within the body in the form of glucose, energy and all. So that fails to respond to the insulin properly. That's how these are the condition. Now, my main target is how we can proceed to get a specific targeted drug. So these are exosomes. Exosomes are nowadays explored for many disorders which have higher potential, which have higher affinity to explore within the research capacity to develop as a potential diagnostic as well as prognostic tools. We can develop these exosomes in a diagnostic approach as well as a therapeutic approach. So these are basically very tiny molecule. These are basically tiny molecules, tiny vesicles which are very small in size, approximately 30 to 150 nanometer in size. And these are importantly secreted from most of the body fluids, including plasma, serum, saliva, urine, amniotic fluid, malignant ascite fluids, and cultured medium of the cell cultures as well. So we can explore the possibilities from exosomes to develop as a diagnostic as well as therapeutic tools or approaches, because these can be further isolated from the body fluid and can be uh, developed or can be loaded with phytonutrients or small molecules to target specific cells and size. The beauty of these exosomes is that these consist of all the essential nutrients, micronutrients, the coding, encoding genes, molecules, and all, because it's a lipid bilayer which consists of messenger RNA, micro RNA, and all the encoding genes with appropriate proteins and all. These are the proteinous in nature, so which has a capacity to develop as a potential therapeutic tools. So what happens is that in patient with type 1 or type 2 diabetic patients, basically insulin regulate sugar level in the blood. So what happens is that when in type 1 diabetic patient, insulin producing beta cells are released, and the protein, they also release the package of protein. That package consists of exosomes. Those exosomes are proteins in nature, so which are taken up by the dendritic cells. So what happens is that these dendritic cells are present with the exosomes, and this exosome filter up the content of these the content of to the T cells, and these T cells have a capacity to work as an enemy to these beta cells. So the pancreatic cells, beta cells, have an argumental capacity that try to keep as a repulsion nature with these T cells and all. So once these dendritic cells are entering within the T cells, the immune cells response is also hurted. So these immune cell responses, T cells take a responsibility and keep as an enemy to these beta cells. So that is the beauty of these exosomes, which could be developed further to in the profound nature of the medications, in the profound development of all the strategy that could be placed with the researcher in drug industry as well as in academia. This could be a best tool to encourage the researchers in the platform of diagnostic. So let me talk about the diagnostic approaches here. So these diagnostic approaches, if we see, this is the rationale behind the diabetics and in the diagnosis process and of the exosomes. So when we see the exosomes, this really consists of many type of microRNAs, messenger RNAs, protein associated with many of the targeted individual targeted genes, and with different proteins binding site, those receptors are recognized by these exosomes that could also be developed in the platform that encourage us to be in the part of the exosomes moieties. These also consist of many different enzymes, proteins, heat shock proteins, cytoskeletal proteins, annexins, binding site receptors at the periphery of these exosomes, because it's a lipid layer. So these lipid raft work as a small molecules and help in the severity of that condition. So these are basically, as we know, these are important and very small size molecule in nanometer size, and contains DNA, RNA, proteins, lipids, and all that makes this exosome as a good tool in the diagnostic approaches. Since we have a capacity to take out those fluid in different from the different region, from different, either from urine, saliva, or from proteins from blood, we can easily analyze these exosomes which are creating havoc to these in the form of dendritic cells and keeping as an anemic stages with the pancreatic beta cells. So these can be this can play a pivotal role. So we have to consider these exosomes in diabetic stages, because this can really helps in better communication between the physician and the patient. So these exosomes they are also easily targeting our different stages of signaling protein pathways as well, which come across as a crosstalk to different platforms. So we eventually have, perhaps we could really correlate these diagnostic approaches and could evaluate or investigate different compositions incorporated within the exosomes. So we get a higher chances of diagnosis, which gives, which can easily give us a biomarkers site which also can helps in boosting up the sites of action that is taking place within the exosomes and could elucidate what is the reason behind the development of this type 1 diabetic or type 2 diabetic condition. So it could really play an important role in the diagnosis as well as prognosis approaches. So we have lots of advantages over exosomes. If we see, these are easily attainable samples and which is non-invasive process. You can collect blood, you can collect saliva, you can collect any fluid easily from the body in the form of from the urine, plasma, blood, or whatever the body secrets. So we can easily capture those easily samples without any invasive process. So these are robust non-invasive process, and these are continuous process which have a long-term monitoring capacity, which eventually helps patient responses, and patients probably get a chance to induce with certain therapeutic agent that over the course of treatment period. And it also, in addition to this benefit, it also reveals comprehensive molecular information. So because of its comprehensive nature of the exosomes, we have a capacity to develop it as a best tool in our research, because it gives a complete and comprehensive molecular information happening due to the molecular events at cellular site. About islet cell functionality, we can easily pick up what is happening at the beta cells or alpha cells functionality in pancreas, what is happening at the inflammatory cytokine site, their mediators, insulin, C-peptide release, insulin secretion, C-peptide secretion, and also on. So we get a complete list of comprehensive information. And there's a, as said, there is a sample diversity for either from blood, we can even take cerebrospinal fluid, plasma, serum, urine, saliva, ascites fluids, and so on, to determine the potential of this handsome research oriented target. So as in the form of biomarkers, this could also be developed. This could develop as a potential biomarker is within the beta cell mass that could lead to apoptosis in type 2 diabetes condition. And there is a big hit to the crosstalk of these microRNA potential as well. So when we talk about the microRNA, that could also be known to be expressed at high level in beta cells. And those specific microRNA could be targeted or could be developed as a targeted specific microRNA for diabetic research. Those are microRNA 7, MIR29A, and MIR146A. These are elucidated as an important and essential microRNA in diabetes research that could be employed in further development of the strategy that could combat as a microRNA tool to approach within the beta cells. So when we talk about high blood glucose level, this could also be altered the component of red pancreatic beta cells. When we go further for preclinical approaches in human trials, prior to that we have to create the potential of exosome in the animal. So for animal research, microRNA 15A is a potential target that we are looking into in our research lab. So rat and human islet cells that releases GAT65IA2 intracellular beta cell autoantigen in human type 2 diabetes and proinsulin. So we come across with microRNA 15A, which have a potential in rat model to develop as a potential exosome tool that could be approached to target the diagnosis in different platforms. And in addition to that, GET65 and IA2 are also shown a promising result that could be helpful at intracellular level in signaling pathways. So application in drug delivery, we could develop these exosomes and can be loaded with multiple drug moieties. Those could be hydrophobic drug, hydrophilic drug, and so on, or intracellular component like DNA, RNA, and protein. Even membrane component ligand and receptor could also be directly targeted within the exosomes or can be loaded up within the exosome to directly target at the specific site of pancreas. So drug could be either hydrophobic, hydrophilic, or genetic material, could be protein material, DNA, RNA proteins, or any of the covalent linkage targeted ligand receptor sites. Small molecules also can be incorporated. On top of that, we are targeting RNA, microRNA 106B and microRNA 222 in rats, which have shown promising effect when we load any of phytonutrients or small drug molecules within those exosomes, purified exosomes, to target the specific drug. So I'm highlighting here the important loading applications that could be placed within the exosomes. Those could be lipophilic small molecules which are passively loaded into the exosomes. And those could be, once we receive the exosomes either from industry or we have developed in our lab, we purify these exosomes with certain processes. Once we have purified, we try to incorporate these small molecules or incubate these molecules within the purified exosome and to develop them as an exosome like vesicles and to have a potential either could be from phytochemical, synthetic agent, or so on. So when these parental cells are loaded within a therapeutic agent, then these are released in exosomes within the body or as reaches within the body that helps in migrating all those composition of therapeutic agent and assimilated within the region. So the another approach could be either transfecting or infecting parental cells with drug encoding DNA, which are then released in exosome. So these three approaches we which we are highlighting here are easy to develop exosomes or load exosome with certain micro essential molecules prior to develop as a therapeutic agent. So this could really plays an important role of the diagnosis as well as therapeutic analysis. So either could be loading parental cells with a therapeutic agent or transfecting parental cells with a drug encoding DNA. These multiple approaches can be employed in the process of developing exosome as a drug moieties. So these are the huge market side that could be taken into the consideration by the drug industry as well. They are trying to approach within not only in diabetic research but in cancer research as well. And at some point we are getting, probably we are getting a good high side so that we can come across with the essential therapeutic with this. So uh, again, in my second half of my seminar now, I will talk about the highlighted research that we are taking in our lab at Taylor's University. So here in exosome research, we can really incorporate even these small targeted molecules to be loaded up within the exosomes and can be specifically targeted to the specific site in the body. So let me talk about the phytochemicals, extracts, and small molecule research, which we are using within the part of diabetes strategy. Recently, we have published this review on another specific target. We recently published in pharmacological research this review as a part of a PhD project, where we have seen that there is a huge potential of crosstalk between toll-like receptor as well as Wnt signaling pathway. So this strategy, this Wnt as well as TLR signaling could also be another target in diabetic research. We can directly target these signaling pathway at intracellular level by creating a molecular events at the site of the approaches and could develop as a therapeutic. So there are many phytochemicals, preclinical drug molecules, many drugs, and even small molecules and peptides have been explored on these signaling pathways, but very few of them have reached to the phase of clinical trials. So in this publication, when you look through, you will find multiple proteinous targeted molecules are shown in this publication, which will give an overview or an idea for a researcher to target these signaling pathways. And importantly, we need to really consider the crosstalk. We need to consider the crosstalk when we apply these Wnt and different signaling pathways at a site of the region. So importantly, Wnt signaling, toll-like receptors, they have a capacity to promote unrestrained adipogenesis and metaflammation. Because of these metaflammation, the pro-inflammatory cytokines, the mediator cytokines like tumor necrosis factor, interleukin 6, IL-1 beta and all, these also create the metabolic inflammation, and adipogenesis process is also elevated. So that could be targeted. If we can slow down these metaflammation and adipogenesis process, we can easily target a essential therapeutic for obesity and type 2 diabetes. So in this publication, if you go through with this, you will find a lot of information highlighting about the targeted therapeutics that could be employed for further research. So in our next target could be endoplasmic reticulum stress pathway, or we could say unfolded protein response. UPR signaling has also shown a promising result within this capacity. So UPR signaling also plays an important role in the diabetic research. In cardiometabolic research, if I say, that would be the appropriate narration in terms of cardiometabolic approaches, because this UPR signaling not only target diabetes condition, this also help in elucidating the targeted approaches within the obesity as well as in cardiac dysfunction. So this could also be explored in a better way so that we can really understand the fact behind what is happening at the different sites. Recently, we have published the potential of arginolic acid, and we have seen it has neither shown any toxic side to these normal animals, nor have any toxicological signs within the normal animals that has in hematological or histopathological biochemical system. So it has been evaluated in our lab for diabetic type 2 diabetic condition, and we are in preparation of the manuscript for this research as well. We have seen that lauric acid, the important composition from we get from coconut oil, has also shown and potential anti-inflammatory effect within the LPS, lipopolysaccharide induced inflammation in rat model. Prior to that, we have concluded by this publication that it has no toxic as well as any kind of alteration in normal animals, neither in biochemical nor in hematological and histopathological studies in normal animals. So it is safe to be consumed as a food additive for further research. And previously we have published the potential role of Centrathera menthalmenticum seeds that have shown to be promising effect on pancreatic beta cells as well as in type 2 diabetic rat model. So this is an important seed which is available in India as well as a Kali Jiri that has potential to be used in multiple diseases, and we have shown that the seed extract, methanolic extract, has a potential role in pancreatic beta cells as well as in different animal model in type 2 diabetic model. Then we have studied the effect of this seed methanolic fraction on chronic type 2 diabetic model for 90 days study, and we have seen that it has not only reduced pro-inflammatory, elevated pro-inflammatory cytokines, but it also reduced oxidative stress, hyperglycemia, and lot of degeneration processes were altered because of these diabetic conditions. All those altered processes were ameliorated. And if you see the it has a multiple composition. It has quercetin, camphorol, flavonoid, decafel, acid, caffeic acid. These are the major constituents, essential markers available in this methanolic fraction. We have also shown protective effect of Vitis vinifera against oxidative stress, inflammation, and apoptosis stages in pancreas of diabetic rat. We have also seen the renacenthine C ameliorates hyperglycemia, hyperlipidemia, and it has shown a promising effect even in oxidative stress condition, which have deliberately reduced or destructed the pancreatic cells. But this renacenthine C ameliorated these all the stages in male diabetic rates. In past, we have published the extract of Woodfordia fruticosa flower. This Woodfordia fruticosa flowers are the essential flower used in Ayurvedic preparation in aswas and aristas in our Ayurvedic system of medicine. So this flower has a potential to reduce beta cell destruction as well as oxidative stress condition in type 2 diabetic rat. And we have shown that the flavonoid have huge potential to be developed in as a medication or as a nutraceutical dietary supplement for the modulation of glucose transporter protein, which is essentially required. These GLUT4, GLUT2 proteins, these are important in glucose uptake, insulin reliability, uptake of insulin, and which are helpful in nourishing or giving energy to the body. So importantly for in diabetes, these are retarded. These are basically affected more GLUT proteins, and also with while taking flavonoids, this could be modulated, or this helps in regeneration process of all those flavonoid could be in combination or with individual composition. And next, in this publication, we have shown that quercetin and cunic acid, when we combine together, it has shown a wonderful effect on liver, kidney, and pancreas tissues in STZ-induced diabetic rat. So we consider these flavonoid and essential phytochemical when combined together also synergize the effect. So we mean to say that some of the multifactorial composition in Ayurvedic drug or in Ayurvedic preparation or as a dietary traditional medicine, we could see when we combine one or two phyto compound together, we can either synergize the effect or we can potentiate the effect of one drug by adding on another uh, phytochemical. So that could be polyphenolic group of compound or flavonoid. So these are the basically quercetin is a flavonoid and cunic acid are enriched polyphenolic group of compounds. So we can combine either polyphenolics, polyphenols, or one or essential uh, uh, group of compound together to get the synergism. We have even seen Pseudovaria monticula bark extract, which has upregulated insulin secretion and reduced downregulated inflammatory cytokines, oxidative stress condition in type 2 diabetic rat model. This vincristine and vinblastine. So vindogentianine is a hypoglycemic alkaloid which we have isolated from Catharanthus roseus and studied on different diabetic conditions. So we have seen in different enzymatic as well as cell culture model, study model, and even on a different animal model to see that vindogentianine has a potential to develop as a hypoglycemic drug. Basically, it is an alkaloid which has a promising effect, which have shown to be effective in many different cancers, but on top of vindogentianine has a potential to be developed as a hypoglycemic alkaloid or a drug in near future if we go for preclinical approaches. So there are different alkaloids which we have isolated from Catharanthus roseus have a promising effect on this drug as a to be developed as a drug or dietary supplements on different diseases model. In our other publication in this we have shown that different multiple phytochemicals can be approached with these different pathways or different drug moieties that could also be approached to be represented as a transporter protein or which could mediate insulin signaling pathway or accelerate the transporter protein translocation. So there will be an appropriate uptake of glucose to the whole body. That is the main intention behind different phytochemicals. So I personally believe mother sorry, mother nature has given us uh, multiple floras, plenty of floras that could be potentially developed as therapeutic agents in different diseases condition. If we can consider those specific phytochemical and test on different platform in different disease model, we can come across with the scientific approaches. There is only the lacking behind the thing is that we only have lack of uh, instrumentation in India, lack of maybe expertise in terms of publication that only have a significant to significant drawback, I would say. If we really consider that, we have to consider phytochemical traditional medications or approaches that could really lead to multiple set of high-end publication that could really help to showcase Indian system of medicine to the global community. And we can conclude that these are the specific or innovative approaches that could be incorporated in different therapeutic agents. So, and within this, we have also shown there is a huge involvement of different mechanisms or approaches which are highlighting type 2 diabetic drugs with different Alzheimer conditions and all, which are showing the effectiveness of anti-diabetic drugs or phytochemicals, which could lead to diminish or alleviate or ameliorate the cognitive impairment, which is the aging process. When a person is heading towards diabetes, there is a possibility that a person could also lead to cognitive impairment. So these Alzheimer process have slow aging process. It has a slow aging process. So we can boost up the strategy of anti-diabetic drug or exosomes targeted approaches or multiple approaches, drug approaches, which could really help a patient to combat many coming across aging processes. So I believe this diabesity, which is a combination of diabetes, obesity, uh, and cardiovascular dysfunction, is a new epidemic. So we have to consider this as an alarming condition for the global community and for the researcher to really develop these um, exosomes or further targeted approaches can be employed to work on a different platform, multiple platform, to reach out to the scientific community or academia or industries to come join hands or in collaboration can be worked with these uh, epidemic disorders. So with this, I would like to end my session. Thank you.
Thank you so much, sir, for your session, Dr. Arya Andy. And regarding the pancreatic diabetes, whatever you have said, it is more informative session. And coming to the second session, I welcome Dr. Satishwaran N. Andy from Malaysia to give a second session. Before that, I request, if it is possible, J. Nagalakshmi Garu to read out the profile of Dr. Satishwaran N. Andy from Malaysia regarding the topic, the second session, which is regarding, just a minute. The second session regarding the in vitro study on anti-neuroinflammatory properties of deoxylefetopin from Elephantopus scabriel. That is the second session topic. I request J. Nagalakshmi Garu to read out the profile of Dr. Satishwaran N. Andy from Malaysia. Thank you.
Good morning to all. This is Jada Nagalakshmi, working as Assistant Professor in Chalapati Institute of Pharmaceutical Science in Department of Pharmacology. A very good morning to all to the one-day webinar session on recent advances in. And it's my pleasure to introduce the second speaker to this session, Dr. Satishwaran N. Andy. Sir is obtained medical science degree from North UK in 2014, and he received Doctor of Philosophy in Biochemistry from University of Malaysia in 2018. Currently, Sir is a senior researcher and development associate in InQ Pharma Malaysia, and he plays a major role in development of new products, preclinically and clinical research, and intellectual property, regulatory affairs, new product commercialization, and post-commercialization support. Now, Sir has a professional experience including working directly with senior project manager and director in developing a new product for current and future products. Thank you, sir. Sir, I invite you to to give the session. Thank you. Thank you, Tony. Thank you, Mr. Tony. I would like to invite Dr. Satishwaran and Andy sir to speak on.
Or sanction whatever is there, fellowship.
Tony, what?
ഇതിപ്പോ ഗൂഗിൾ ചാറ്റ് എന്തത് ഓഫ് അല്ലേ മൈക്രോഫോൺ ക്യാമറ ഓഫ് അല്ലേ
ഇവിടെ ഉണ്ട്.
Tony?
Sir Satishwaran sir, you can continue. Sir, you can continue. Sir, I am not able to hear you, sir. Satishwaran sir, you are visible, sir. You are visible, but your audio is not. Your audio is not good, sir. You are visible. Sir, you are visible, but your audio is not. Your audio is not good, sir. You are visible to me. I am not able to listen your audio. You are okay. You are okay. You are visible to me, but your audio is not coming, sir.
Unmute the video, unmute the audio.
Hello. Sir, are you able to listen me, sir? But your audio is not good, sir. Please unmute it.
You can also unmute his audio.
Admin also can unmute.
Tony, you can also unmute this audio.
Sir, can you able to talk directly through lobby, sir? Is it possible for you to remove your earphones? Yes, yes. Thank you. Sir, your audio is in mute or unmute, sir. Please check it. Hello. Sir, here it is showing your audio is in mute, sir. Here it is showing your audio is in mute. You just try to unmute it. Sir, Dr. Kalarajan, sir. Hello. Sir, Dr. Kalarajan, sir. Hello. Hello.
Or otherwise, you can join VPP sir to take the
Hello.
session. Tony.
Sir, Pandey, sir, are you ready to take the session, sir?
Yeah, I am okay. Yeah.
Yeah, you just start, sir. Dr. Satishwaran, sir, will be joined in the next session, sir. Thank you.
Okay. So, am I audible?
Yes, yes, sir.
Okay.
You just on your video and you can proceed, sir.
Okay.
Yes, thank you, sir. Pandey, sir, you can continue.
Okay.
You can share your screen.
Okay. Good morning to all. Yeah. I am Dr. Vijay Bandi from Chalabri Institute of Pharmaceutical Sciences. So today I am going to give talk on the art of scientific writing. Am I audible?
Yes, yes, sir. Sure.
Okay.
You can proceed, sir. Thank you.
Yeah. Okay. So here, writing I'm saying art of writing. In fact, the writing itself is art. Okay, so if you talk about the scientific writing, so there are different way of writing the scientific papers. Suppose in case of writing, suppose we give some concept to few people to write that concept, everyone will come up with different write-up. If you go through the write-up, you can find a lot of difference in the way they write. Okay, so some of the write-up is very interesting to follow and read. Some of the writing is not really very interesting to read. Sometimes you might stop in between and you go away from that reading that the write-up. So it is an art actually. So the how we are writing that flow is all very important. In case of if you talk about the scientific writing, so scientific writing in the different form. Suppose as a student, someone you want to submit the assignment. So there is a protocol how to write the scientific writing. So if you write the assignment or you submit a research article to the journal or if you submit a review article to the journal. So these are kind of scientific writing. So what is the purpose of writing the scientific paper? As we know, I mean, the main purpose, in fact, if you see some 40, 50 years before, people will do research and they publish the article. The purpose is to disseminate the knowledge to the other scientific society. Okay. So that's the main idea of writing the scientific paper and publishing. And other important thing regarding writing scientific paper, normally the write-up is reviewed by the peer. Suppose you have some research article, you prepare some manuscript, and if you submit to some journal, so that is reviewed by the expert in that particular field, and they will comment on your work. So that is the main important other reason why we write a scientific paper. Apart from that, the purpose is to advance the science. See, for example, here I have given some picture. If you see the basis of some, the aeroplane, Wright brothers, the one what they developed, might be 100 years, more than 100 years before. But now you see the actual one. So there is a lot of transformation from the actual one to the advanced one. So this transformation will happen by continuous research. So anyone, if they do some kind of research, they're adding some knowledge to the existing knowledge. So very simple thing. So they do research to add some knowledge to the existing knowledge. So that way we are building the science and advancing the science and that way what we are now. Another important purpose of writing the scientific paper, as we know, this commonly we everyone agree for the purpose of graduation. Suppose normally say some universities they say if you want to graduate as a PhD, you need to have at least two science citation index journals, or if you have masters, at least one paper. So this kind of guidelines are given. So for that purpose, someone they will go for publication. Apart from that, suppose those who are in academic field, they want to get promotion. Suppose from assistant professor to associate or associate to professor, they need to show some kind of publication. Importantly, so the person will get recognition in the particular field. Suppose someone is working on neuropharmacology and someone is working on metabolic disorder. So if he continuously work on that particular field, people will recognize that particular person from that particular field. So this kind of recognition will be uh, 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 obtained. I mean, okay. Now, before going to write any scientific paper, first we should know about our audience, to whom we are address our writing. So first important thing, who is going to review our write up? Most probably if it is assignment, might be your supervisor. If it is, if you submit to some journal, it might be the referees or reviewers. Actually, the reviewers play an important role to judge your writing. So normally the important role of reviewer, the reviewer will help the editor's decision to accept or reject the paper. Actually, sometimes even the reviewer made comment and they suggest for accept the paper, but the comments is again reviewed by the editor. to decide whether the paper can be accepted or rejected. Another important thing, the role of reviewer. So mainly he will evaluate the paper or the manuscript, whether it is scientifically sound. Importantly, there are two important things in the paper, research paper. First thing, they will look for the research hypothesis and research methodology. Suppose someone is doing some natural product. Okay, this particular natural product has been reported for antibacterial, antiviral, anti-diabetic activity, and so on so activity. So there was no study on, say, antipsychotic activity. So I want to do the antipsychotic activity. Suppose if the person is claiming or giving the hypothesis this way, normally the scientists won't agree. So in case the person, suppose I want to study for antipsychotic activity of this medicinal plant, what is the rationale for choosing this particular plant for this particular activity? This is the key point should be given, especially in the introduction chapter. So this is a research hypothesis. And other thing, the research methodology. So if the study design is well designed, then there won't be any problem in the getting paper published. So these are the two things any researcher should give importance actually. Other thing reviewed by the reviewer is whether the work is original. Actually, nowadays we have the software to analyze whether your write-up is original or not. We have the different software like Turnitin. Suppose if you put your write-up in the Turnitin, it will give the similarity index. So normally the journal will allow your similarity index should be less than 20%. If it is more than 20%, normally they don't accept your paper. So your write-up should be original. Okay, that's very important thing. So this normally in the editorial team, they will, if you submit any journal, sorry, any paper to any journal, so the editorial team will subject your write-up to that software, and they find whether your write-up is original or not. If it is not original, sometime from that desk only they will reject your paper. And other important thing, novelty in research finding. Actually, here two things we need to see actually. Normally, if you, there are two type of journals. One is subscription-based journal. Other one is open access journal. Subscription-based journals, normally they are looking for the novelty in the research finding because they have the limited space. So if your work is really novel, then only they will admit your paper. Whereas in open access journal, so they mainly focus on your research hypothesis and methodology. Your research design is good and your research hypothesis is valid, then they admit your paper. So the next thing regarding the readers. So readers, that is our target. So who is going to read your paper? So normally the readers, not necessarily they are from your expertise in your field of research. Most of the time they are more or less knowledge about the paper. So there are two key points they look for. Some people they want, suppose someone published the work on antipsychotic activity using some different animal model, and other researcher he want to test some their compound for antipsychotic activity. So they want to follow your research protocol. So then they will follow your research methodology. So that is the purpose of reading your paper, to look for the research methodology. There are some people, they look for the research finding. Suppose this plant is having some antipsychotic activity. So you are the first person who are claiming for that particular activity. Then people will see, okay, this plant is having this particular activity. Why don't I take this work to further in the development of novel drug discovery? So this is okay. Next, we should know about the journal. What journal you are targeting. Normally, there are normally they categorize into two categories like index journal, non-index journal. So if you talk about index journal, so normally we give importance to science citation index. It is comes under Clarivate Analytics. Previously, it was with Thomson Reuters. Okay. Another one, science citation index expanded. So if you publish your article index in this particular category of journal, then your article will reach to higher level. Then apart from that, you have Scopus and Embers covered by elsewhere. And you can also see whether the journal is indexed in PubMed. Normally, as we know, suppose someone want to do some research, if you want to do some literature search, normally what they will do using the keyword, they search in the PubMed. Okay, so if the journal is indexed in PubMed, your article will appear in the PubMed. Then people can access to your work and they can use your work for the research. Then the last one, Google Scholar. I think Google Scholar it covers most of the journals. I mean, so most of the journals is covered in Google Scholar. Sometimes some journals not covered in any of these things, but it might cover in Google Scholar. So if you want to choose before going to choose the journal, you need to go for that journal website and see this particular journal in this indexing what type of indexation like science citation index or Scopus or PubMed. So you can give this priority for this three things. So here they have given some the difference between Embers and Scopus. Here Embers focuses on the full text indexing of biomedical content, bringing insights through structured indexing of the content. Whereas Scopus focuses on abstracts and citation, enabling navigation of the published literature. Okay, this is other important thing. Okay, suppose I said science citation index journal. But if you go through the website of the journal, many journal falsely claim their journal is indexed in Science Citation Index. How do we know whether the journal really indexed in Science Citation or Web of Science Group? So here I have given that website here, clarivate.com. Okay, so if you go to this website, this web page will open. So here you can type your journal, your targeted journal. Suppose here I have given my targeted journal is experimental animals. Suppose I type the experimental animals here and I search. So I got that one. It is already in the Web of Science. So that means definitely I can choose this journal for publication. Sometimes some journal they say it is in Web of Science, but that journal name, if you give here and if you search, you will find zero result. That means that not in the Web of Science. So better you can avoid you publishing that particular journal. So already I mentioned there are two types: on subscription-based journal or traditional journal. So here readers are required to pay for the content that read. Suppose you see some elsewhere journals, and if you want to read some article, if you click that particular article, it will ask to pay money. So if you pay, then only you can access to that full article to read that one. So that way the readers will pay for your article. Another important limitation, the traditional journal, it has a limited space for publication. I mean, the rejection rate is very high. So suppose one journal, one issue, it can able to publish only 300 articles. But sometimes that particular journal, it might receive 3,000 articles. Okay. So what they will do, they try to reject maximum. Sometimes some journal will reject up to 90 to 90%. So we cannot say the rejected journal is not in good quality manuscript. Okay, so the rejection based on the priority. Okay, so this is the drawback of publishing your article in the traditional or subscription based publication. So normally here they are looking for the novelty in your finding. So you need to do extensive research and you need to in the finding. So then only they will give the preference. So most of time, this kind of journal, suppose in elsewhere journal, immediately they will reject. Sometimes some of my friends say, Pandey, I submitted last night. Next day morning, I found my paper got rejected. Okay, so I was really disappointed. Actually, the thing is here they go through the abstract and how much work you have done, and then only they decide. Actually, in the first attempt only they decide whether to admit your paper or not. So the editor desk only they will reject your paper. So that is the drawback of subscript journal. And the other thing is here. Normally the traditional journal they have the page limitation. Most of time they will allow to publish only 8 printed pages. Sometimes if your length of paper is higher than this record pages, suppose you have some 12 pages, printed pages, so remaining 4 pages you need to pay. Sometimes the remaining 4 pages, the charge will be higher than the published in open access journal. Actually, I got one of my friends, he got published in, got accepted in elsewhere journal, and unfortunately his the printed pages, it reached around 12 pages. So the elsewhere asked to, the particular journal asked to pay the excess page. So he calculated that came in very big amount. So sometimes you need to decide, suppose your length of paper is a bit higher or very bulky, better you can go for open access journal instead of going for subscription based journal. Another important thing, the copyright of the published content usually transferred to the journal. So while submitting your paper, you need to give the agreement. So you need to transfer your copyright to the particular journal. And regarding open access, very good thing here, anyone can access to your article, and the reach is very high and you will get more citation. But the difficult part here is they charge the authors, the article processing charge. Okay, so sometime the article processing charge, it might reach up to 3,000 US dollar. See, like in Asian country, suppose in India we have suppose 70 rupees in US dollar. So if you multiply with 3,000 USD, it might reach around 2.5 lakhs. Okay, for publishing one article, 2.5 lakhs. I don't think we can offer that much amount of money. Okay, so that is a very big limitation for the researchers, especially from the Asian countries. But the good thing, the space for publication is not limited, so they can any number of pages they can publish and freely available to the scientific community. An important thing, the citation is very high. If you go to the Western countries, those universities, they suggest or recommend the scientists to publish in the open access journal because their article will reach to high extent and they get more citation when compared to subscription based journal. Okay, now let us see the structure of paper. So normally, as we know, this is the following structure we follow, like abstract, like introduction, materials and methods, results and discussion, and conclusion. So abstract might be structured or unstructured. So they allow less than 250 words. The introduction. So introduction is very important actually. That will motivate the readers to go through your paper. So normally they look for why you want to do this particular study. Okay, so they will look for that one, and that will motivate the reader to read further. And you also describe what are the work you have done in the particular paper. So that also you will describe. So based on that one, they will decide to read further or not. Another important thing, materials and methods. So we need to provide sufficient detail. What are the methodology you have used, especially to reproduce the experiment. This is very, very important. Another the thing here, if you go for the similarity index or plagiarism check, most of the time we get more similarity index in the materials and methods section because we follow some protocol from some other journal. Most of the time we use the words for our article. So if you analyze your paper with the software, it will give more similarity index, and especially on this part. So here better we need to rewrite on our own whatever way you have done the work. So that way if you write, I think we can avoid the similarity index in the materials and methods section. Other thing, results and discussion. So normally it combined together. So here we need to present the data in the form of like figure or tables. So most of the time it is combined. So last but not least, the conclusion. So some people, they look for abstract introduction. Finally, they go directly go to the conclusion. So here they present the outcome of the work. I mean, what is the final finding? So here normally we should avoid any hypothetical conclusion. I mean, whatever the outcome you got from your research, you need to mention only your your finding, not like any hypothetical statement here. So this will motivate other researchers to continue other work. So now, so yeah. So in the introduction, so we need to stimulate the readers about the importance of your work. So normally they use some kind of terminology here to stimulate the reader. Like in recent years, suppose someone is looking for drugs to treat COVID-19. So they can say in recent years, many people are affected with COVID-19. So it's alarming. So there is a need for the drug. So this kind of stress if you give, people will get interested to read further. So, like need for your research, like emphasize the contrast between actual and desired situation. The desired part of the need can be combined with the task in the single sentence. This sentence expresses first objective, then action undertaken to reach the objective. For example, to confirm this assumption, we studied the effects of a range of inhibitors of connection canals. So these are some examples they have given how we need to combine the objective and the task. For example, here to assess whether such multiple coil sensors perform better than single signal one. So in the task, what the others have done from in other words, the task clarifies your contribution, your objective to prepare the reader to. So here they have given some example. Use whoever did the work. Normally, you or your colleagues as the subject of the sentence. So for example, they use we or perhaps the authors use a verb expressing the research action like measured, calculated. So these are the things we need to follow while writing the introduction. So these are some verbs, they express the research action. For example, we assess the effect of larger doses of, we calculated, we compared, we computed the velocity predicted by, we derived the new set of rules, we designed a series of experiments. So we determined, so this way you can narrate your statement in the introduction. We develop a new model, or we evaluated the efficacy and biocompatibility. So these are some examples we can follow. And importantly, we need to mention about objective of that particular work. So use the document itself as a subject of the sentence. For example, here, this paper clarifies the role of CXXC on calcium oscillation in neonatal cardiac myocytes. Otherwise, you can say this paper presents the flow effects induced by increasing the hepatic artery pressure. So these are some examples how to mention your objective. Yeah. So this paper clarifies the role of soils. This paper describes the mechanism by which this paper detailed. So these are some examples how to mention your objective. Sorry, I just take one. Now let us see about the materials and method. Important things should not be bored to read. So to make the section interesting, you can say explain the choices you made in your experiment procedure. what justifies using a given compound or concentration dimension, what is special in your methodology when compared to other people what they use. So these are something you need to stress and make the readers to get some interest on reading your procedure. Okay? So if you use a standard or usual procedure, like mentioned as it is, so do not make the readers guess. Make sure the paragraph's first sentence gives them a clear idea of what the entire paragraph is about. Actually, this is true for the entire manuscript. So normally sometime we will divide the manuscript in the different paragraph. So at the beginning of the paragraph should say what the content of the particular paragraph. We can say it is like abstract of the particular paragraph. So then only the reader will decide, shall I continue the paragraph or I can jump to the next paragraph. So this is true for all sections actually. So consider using some table schematic diagram. Suppose you have some study design, you can make the study design the schematic diagram so that people can easily follow your protocol. Other important thing already I mentioned, make sure especially the materials method should have the less similarity in text. Okay, so normally you can run the Turnitin, then you can get the plagiarism report. So results, now let us see about the results and discussion. Results normally present the two form, figures or tables, but sometimes some people, they will present the data both figure and table. It should be avoided. Either the data should be present in the table form or figure form. So you need to decide which form is suitable for the particular data. For example, you have some kind of quantitative data like biochemical parameters or hematological parameters. I think better you can go for tabulated form so that people can even know the value of the normal value and the abnormal value, all the things they can understand. Some other data you can go for figure format. So normally research and discussion sections are combined because results alone don't make any sense. So we need to combine together. So when reporting and discussing your result, do not force your readers to go through everything you went through in the chronological order. Instead, state the message on each paragraph upfront. Already I mentioned. So any paragraph in the manuscript and the first sentence should describe what the entire paragraph about it. Okay, so then only the reader will decide to read further or not. Okay, do not simply summarize the points already made in the thing. Okay, and so whether or what extent you have succeeded in addressing the area. So at the here we are talking about conclusions. I covered here. Okay, at the end of your conclusion, consider including perspective. I mean the future perspective or future planafor. That is an idea of what could not should be done and related to the present work. If you include perspective, clarify whether you are referring to firm plans for yourself or your colleague. For example, sometime if you see in the conclusion, studies in this direction are currently underway in our laboratory, or sometimes they say to an invitation to readers. One remaining question is, so leave it to the other researchers to continue your work. Okay, now I talk about the abstract. So why I give abstract at the end? Actually, while writing the research paper, first you need to start with introduction, materials and methods, research and discussion, and conclusion. Finally, you need to write abstract. Then only you will get the clear idea actually. Then normally abstract will come first before introduction. But while writing your research paper, you need to write the abstract after complete all sections. So typically the readers are mainly interested in the information presented in the paper's introduction and conclusion section. Primarily, they want to know the motivation for the work presented and outcome of the work. Then it might want to know the details of the work. Actually, abstract section plays an important role to attract the audience. Suppose very good abstract will attract the audience to read further your work. So in most of the journals, they try to limit the content of the abstract so that you cannot give, especially the subscription based journal or traditional journal, they are very particular in the limitation of the words in the abstract, because they don't allow you to give more information on the abstract. In that case, the reader might not go in detail in the full manuscript. So they limit you to write very little information on the abstract. But if you want to get very good citation for your research, better you can try to give as much as information in the abstract. So sometimes some people cannot access through the full article, then they will try to get the information only from the abstract. So that case, if you give detail or more information in the abstract, so that information might help to cite your article. Okay. So now, okay, we prepared our manuscript. We need to choose what journal we can go for. So already I mentioned, you remember there are, you always target this whether the journal is indexed in Science Citation Index, PubMed Index, and Scopus. So if any journal indexed in three, I mean indexation, then you can definitely you can go for publication. So how to find whether already I mentioned the journal website will give our journal is indexed in so and so, but you need to double confirm. You can choose that journal and search in that particular website to find whether the journal is indexed in the particular site. Then only you can decide to go for the particular journal. So these some guidelines. Okay, you want to publish your article in elsewhere journal. Elsewhere they have thousands of articles. So which journal I can go for? So there is one option is given in elsewhere website, like journalfinder.elsewhere.com. So here you can give your title of your paper, your abstract, and you can give some keywords, and you can I mean search the option. Then it will list out all the possible journals. Then from the possible journals, you can choose the right journal for your paper. Similarly, you have option in other group of journals, for example, Springer Nature. So here they have journals suggested. So here you can do manuscript title and manuscript text, and you can search for the right journal. So it will give the suggestion. So based on the suggested journal, you can choose. Then you can submit your article. So this will help you to avoid the direct rejection of your article from the particular journal, because sometimes if you submit, you do a lot of work. I know once you start to prepare your article to the particular journal format, you need to do a lot of work, the formatting and other things. But once you submit the article, immediately they say your article is not under scope of our journal, so better you can find other journal. Okay, so to avoid this kind of delay or your work, you can so you can finally you have journal finder in village group of journals also. So this way you can search your journal. And for writing the article, suppose you are writing your article, so we need to use the reference formatting. So better you can choose the software. So this will help to format your reference automatically without any problem. So those days we people we do our research some maybe 20 years before. We need to type each reference manually. But now we have the software, like you can go for EndNote software or Mendeley software. Actually, EndNote software is a subscription one. I think you need to pay. I mean, institute need to pay to procure the software. Then only we can use it. Whereas Mendeley software, it is a free version. You can download it and install in your system. Then you can insert your reference to your manuscript using this software. So this will help you. Okay, I give some brief idea how to insert that particular reference in your manuscript. Suppose I want to insert this article like antipsychotic like activity of noni in mice. So what you can do? So I search. I mean in PubMed search I did antipsychotic and noni. And I got this article. So I want to include or incorporate this reference to my manuscript. What I do here? I will, there is one option given here is send to. So if you give send to, it will list like clipboard, my bibliography, collection and citation manager. And if you click citation manager, it will choose already, suppose if you install EndNote or Mendeley, it will go to that particular reference software and it will install in the I mean, your paper. So normally in the citation manager like Mendeley or EndNote, it will form some kind of library. So once you insert that one, it will go to that library. From the library, you can extract to your manuscript. So in case of Science Direct, you search and you got some paper, you want to export this paper. So here they have given export. So here save to Mendeley. So if you click here, it will directly go to the Mendeley, your library, and from there you can incorporate the reference in your manuscript. Or you can go for export citation to RIS. So this will normally go to the EndNote. Okay. So this way you can insert your citation in your manuscript. So no need to manually type all the references. Another important thing, the language check actually. So definitely all we are not, I mean, expert in the English language. So this kind of software can help actually to edit your manuscript. For example, Grammarly, Typely, like Ginger. So most of these, I mean, software are free. But if you pay, it has more options. For example, some software, it helps to rephrase the sentence. Suppose you have some write up and you put in the Turnitin and it show the similarity index highlighted. So if you want to rephrase the sentence, if you have the original software, this one, if you give that sentence, it will rephrase the sentence for you. Then later you can do some minor editing on that sentence and you can use it in your manuscript. So this software help to write your article. Other important thing actually before the journal do your I mean similarity index, better once you complete your write up, you can use any of the software to find what is the similarity index. So you make sure the similarity index should be less than 20%. If it is more than 20%, better you can rephrase the sentence and make it less than 20%, then you submit to the journal. So you can use like iThindicate, Turnitin, Grammarly. So these are some of the software you can use and to find your similarity index. Okay, suppose you submit your article. Other important thing, how reviewer look your article. So this is the, I mean, the way you look your manuscript and the way the reviewer look your manuscript might be different. And so that is the reason, I mean, sometimes you might fully satisfied with your manuscript, but once you see the reviewer's comment, you really get shocked actually. And even you cannot imagine, and sometimes the reviewer comment make your work cannot be published anymore. For example, here I have given some example here. See, there is one paper, they studied anxiolytic mechanism of action and quantification of this particular compound using animal models. So the reviewer made comment, the authors attempt to establish the positive mechanism action of this particular compound as an anxiolytic activity using the animal model. Okay. So the major comment what they made here in the mechanistic study to establish benzodiazepine GABA A receptor mediated mechanism, the authors used flumazenil benzodiazepine antagonist interaction study. Flumazenil is a competitive antagonist, I mean, at the benzodiazepine GABA A receptor. Like naloxone, it has rapid onset of action within 5 minutes and short half-life. Half-life only 1 hour as per this reference. Rodents are fast metabolizers when compared with humans. Based on this study protocol, flumazenil was administered 90 minutes before the behavioral task. After 90 minutes, there might not be any flumazenil effect. Therefore, flumazenil supposedly should be given 15 to 20 minutes before the behavioral task, as described in the particular reference. So, moreover, the positive control diazepam should be included to show the flumazenil reversal action. You see here, they have done excellent work, lot of work they have done, but unfortunately here there is a problem in the study design. So flumazenil was given 90 minutes before, but supposedly it should be given 10 to 15 minutes before for the behavioral study. Okay, so because of this major reason, the reviewer reject the paper. So now very difficult to rectify it. So most of time the rejection of the paper not mainly because they are looking for the novelty of your finding, they are looking for the study design. So you need to very carefully do the study design, and sometime what happened, this 90 minutes before, not only me, some other person already they reported in the literature. So here only we need to look for which journal you are looking for. So if you refer for some standard journals, so the standard journal have the standard reviewers, they review the article uh, perfectly and make the uh, work publishable. So if you follow that particular published article, you might not have the study design problem. So I can always suggest my student to follow the very standard research article to follow the study design. So if they do some error in the study design, and very difficult to rectify it. So this is a very simple example. So you see they have done a lot of work, but because of the problem the study design, they are not able to publish it. Okay. So not only here, even this work they send to any other journal. Because of that reason, they will reject this paper. But unfortunately, there are some predatory journals. So if you pay money, they will publish your article. So if by chance these authors, okay, they try to different journal and they are not able to get the paper published, then what they will do? There are some predatory journals. If you pay money, they will publish it. So they will send this article and happily they will publish it. But unfortunately, the people who follow this article again, that will have the negative impact to the scientific community. So better we can follow the, standard journals article so that we can follow the good study design. Okay, so this one other thing, suppose you submit the article to some journal and how to respond it, that is also very important. Sometimes they give the option to the authors, how they are responding to the reviewers questions. Based on the response, they will decide whether the paper can be accepted or not. Suppose for the here I have given some example. So how they are responding, to the reviewers comment. So just we acknowledge the reviewer. First and foremost, we would like to thank the reviewer for the constructive comments and suggestion for the improvement of this manuscript. Okay, so this way we are appreciating the reviewer. Then let us see the comment what the reviewer made for this particular article. In section 3.2, the effect of so and so on expression of meth-induced CPP in mice, the authors report that a repeated measures ANOVA yielded no significant results for treatment and treatment trial interaction. However, the authors have again conducted the post-hoc tests on non-significant results, and it is not statistically valid to probe the treatment effect when p-value is 0.3699. As a result, the authors cannot state that mice displayed a preference for meth or the compound attenuated meth-induced CPP. It is possible that the particular compound does not reduce place preference for methamphetamine, but that the study is underpowered to demonstrate this. See here, they are questioning the statistical analysis report. Okay, so if we not properly address the reviewers' comment, definitely this paper getting rejected. So see here how the author responded to this comment. Thank you for the reviewers' comment. Just they acknowledge the reviewers' comment. So I mean reply to the reviewer. Though the repeated measures ANOVA yielded not significant results for the treatment and treatment trial interaction, it showed a significant result for the trial. So the post-hoc Panfroni test compared the data between preconditioning versus post-conditioning. So here the time only the important factor. So thereby this particular meth at 0.5 mg per kg showed significant difference respectively. So the description of these results is revised according to thing, and the changes have been highlighted with this blue font. So here we address the reviewer comment, and the reviewer also accepted this comment and approved the publication. So addressing the reviewer comments also very the important task for the authors. So listener by practice they will learn it actually. So carefully we need to go through the reviewer's comment and we need to properly address it. Okay, here I would like to conclude my presentation. So I would like to express my sincere appreciation to Sir for giving the research grant to our Chalabri Institute of Pharmaceutical Sciences. I would like to express my sincere appreciation to our board principal, Dr. Ramarao sir, and our board chairman, Sri Y.V. Anjaneya Garu, for continuous encouragement and support for our research. My special thanks to Prof. Dr. Narendra Babu sir and through Isha Tony and Nagalakshmi madam from the Department of Pharmacology for the tireless effort to organize this webinar. Last but not least, I would like to thank my friends, Dr. Aditya, Dr. Kaliraj, and Dr. Sadishwaran to accept our invitation to participate in this event. Thank you, one and all.
Thank you so much, sir, for your presentation on the art of scientific writing. I think most of the people have a very good work in their hands to publish. They'll do a very good, perfect, potent research, but actually they don't know how to publish the article because they have a lack of their confidence as well as due to financial crisis, and also their problem to choose which article, I mean, which journal is best for them to publish their work. So I think I hope most of your presentation through your slides have given them clarity, and your presentation may be helpful for them a lot. I think so, and thank you so much, Dr. Pandi sir, for this session. And now I request Dr. Satishwaran sir, who is a Senior Research and Development Associate INQ Pharma from Malaysia, to take over this session. And I'll hand over to Dr. Satishwaran. Sir, are you in online, sir? Dr. Satishwaran, sir? Yes, Dr. Satishwaran, sir, I can able to see your PowerPoint, whatever you have shared. Sir, Dr. Satishwaran sir, are you able to hear me, sir? Requesting all to be patient, and Dr. Satishwaran is coming to online and his audio is through WhatsApp call. So be please patient. They'll be back soon. Yes, sir, you can now continue, sir. Yes, sir.
Yes, sir. So I can go on. So can you hear me, right?
Yes, sir, you can continue.
Okay, hello everybody. Today I'm going to present on part of my research, which is in vitro study on anti-neuroinflammatory properties of deoxyribonucleic from hereditary stable. So before I go into details, I would like to give a brief introduction background on neuroinflammation. So as we all know that neuroinflammation is a defense mechanism linked to protecting central nervous system. But recent studies have shown that inflammation in brain is capable of causing a neurotoxicity and also neuronal cell death. But the major cells that involve in neuroinflammation are microglial cells. So microglial cells, we can say that they are the macrophages in the brain. So uncontrolled and overactivation of these cells will eventually activate various cells in the pathway that will lead to production of various inflammatory mediators, cognitive environment, and amyloid beta protein accumulation, which contributes to neuronal cell death and also neuronal cell injury. So the current available drugs for neuroinflammatory disorders are very symptomatic, that they can only elevate the symptoms, but the efficacy on the preventing the glial cell activations are rather compromised. So therefore, one of the natural compound, which is deoxyribonucleic with a novel molecular anti-neuroinflammatory target site, that will lead to a development of an effective therapeutic agent for neurodegenerative disorders. So here right now is a very little background on how the microglial cells actually works. So in inflammatory condition, the various of cytokines and chemokines, which will further activate the other inflammatory cells such as astrocytes. Further, the astrocytes will go through astrocytosis. They will produce more cytotoxic molecules, which include cytokines and chemokines as well. That will eventually lead to neurotoxicity and neuronal cell death. So upon this cell death, neurons will release all inflammatory cytokines and chemokines, which will eventually go back and activate the mitochondrial cells. So this process also known as a feedback mechanism. Yeah? So in my research, I basically focus on how to dampen or reduce the activation of the mitochondrial cells by external stimulation. So now we are going to look at the compound that I used in my experiment, which is deoxyribonucleic. Deoxyribonucleic is a therapeutic compound from Ericothapus capa plant. So Ericothapus capa is an aromatic plant. We can found it in tropical Africa, Eastern Asia, Indian subcontinent, Southeast Asia, and Northern Australia. So these plants consist of four major bioactive compounds, which includes deoxyribonucleic, isodeoxyribonucleic, scabatopin, and isoscabatopin. But previously, there has been a lot of research has been done on extract of this plant, which is on membrane power, wound healing activity, apoptotic activity, and also anti-cancer properties. But in the compound itself, there are only fewer studies has been done on compound, which is on anti-cancer activity, but also hepatopoietic activity. But there is no research has been done on anti-neuroinflammation. So that brings to the objective of my research, which is to determine the anti-neuroinflammatory activities on EEG against alveoli-stimulated mitochondrial cell, and also to evaluate the anti-neuroinflammatory activity influencing pro-inflammatory factors such as cytokines and chemokines, and also to elucidate the molecular mechanism of action of creating alveoli-stimulated mitochondrial cell, and finally to validate the signaling pathway that involved in NF-kappa B translocation. So as we go on with the method, so usually we plant the plate the cell and we pre-treatment with EEG that induces the LPS. So our first experiment we conducted the cell viability assay to study the toxicity of the compound. So from here we can know we determine the working concentration, and further into our second analysis are the two major pro-inflammatory mediators assay, which is nitric purification assay and also prostaglandin E2. And thirdly, we also perform a protein array analysis to study the cytokines and chemokines expression by B2 cell. And finally, we also perform the vesicle analysis and study the molecular mechanism and also a different expression of proteins by the mitochondrial cell. So moving on to the results and discussion. So this is the results for the cell viability assay. From here, what we can conclude that EEG does not cause any toxicity in this concentration, which is from 0.53 micromolar to 2.5 micromolar in B2 mitochondrial cell. Along with that, EEG also protects you cells against alveoli-stimulated toxicity in a dose-dependent manner. So therefore, we use concentration around this range for experiment. Next, moving on to our results on NO and PG2 production. So this is the result for 91 assay on from and also on prostaglandin E2 cell. So the reason why we measure these two molecules because nitric oxide and prostaglandin E2 are the most potent signaling mediators which contribute to the initiation of neuroinflammation. So as our results suggest that the EEG suppress NO and PG2 production in a dose-dependent manner compared to the treated group. So this is the schematic diagram shows that EEG is able to suppress the NO and PG production. So don't worry much about this image. I will complete this diagram as we go on with my presentation. And next, we perform the vestibular analysis analysis on iNOS and COX-2 proteins. So these are the two proteins crucial in synthesizing NO and prostaglandin E2. So they are directly actually correlated. So this is our result for iNOS and COX-2 from reservoir analysis. So our results suggest that we will be able to suppress iNOS and COX-2 in a dose-dependent manner compared to LPS medium group. So once again, we can see here that the suppressive activity of PED in iNOS and COX-2 further suppress the expression of and production of NO and PED2 production. So in order to study the molecular mechanism underlying the anti-neuropathy effect of the EEG, we performed a Westergaard analysis on mitogen activated protein kinases. So as we all know, that MAPKs are one of the major cell signaling pathways that involve inflammatory condition. So this is our result for our MAPKs. So results here suggest that EED suppress all three MAPK expression, which includes PWK, EK12, and JNK in a time-dependent manner compared to LPS treatment group. To further check this pathway more using all three MAPK pathways inhibitor. So as you can see in this slide, SP202190 is an inhibitor for PKD pathway, SP600125 is an inhibitor for JNK pathway, and U126 is an inhibitor for ERK pathway. So based on our results, so this result further proves that and confirms the involvement of MAPKs modulating the effect of EEG mechanism of action. So once again, we go back to the diagram. The diagram further shows that blocking of this pathway will eventually shows the suppression of iNOS and COX-2 expression. So next, further we study the IKB phosphorylation and also NF-kappa B activation. Whether the NF-kappa B activation is PI3K dependent. So this is the result for IKB alpha phosphorylation and also NF-kappa B activation. First, our results suggest that we are able to suppress IKB alpha phosphorylation and also NF-kappa B translocation in a time-dependent manner compared to the LPS treated group. And next, to check their pathway, its dependency on AKT pathway on IKB phosphorylation and NF-kappa B translocation, we performed Westergaard analysis using both PI3K and AKT pathway inhibitor. So this is the result of it. So the reason why we study this because PI3K pathway is the one of the pathways that are crucial for inflammation, and they are actually closely associated with the to further validate the NF-kappa B involvement in iNOS and COX-2 production. We also perform Westergaard analysis of once again in iNOS and COX-2 along with NF-kappa B translocation inhibitor, which is ethyl 3,4-dihydroxysteamine. So from here we can further confirm that NF-kappa B translocation is crucial in the mechanism of action. So once again, in this summary, we can see that inactivation of PI3K pathway results in suppression of both iNOS and COX-2 expression here. So further, we also perform check whether the DT bond with the AKT pathway as well. So AKT is another pathway, another downstream pathway to PI3K pathway, which is also crucial for inflammation condition. So this is a result for AKT phosphorylation. So our results suggest here that DT able to suppress AKT time-dependent manner compared to the healthcare student group. To further confirm its involvement in INOS and COX production, we also performed analysis on the INOS and COX along with the AKT market. Further suppressed them and validated the involvement of AKT in INOS and COX production. So once again, we go back to the summary. Then we had the AKT pathway between PI3K and NF-kappa B. So we can also see that suppression of AKT also leads to suppression of NF-kappa B, and further it will result in reduction of INOS and COX. And finally, we also perform protein array analysis on cytokines and chemokine production. And also we study whether their production is through PI3K, AKT, and also MAPK pathways. So this is our result for the protein array analysis. So the results suggest that we will be able to suppress all the major pro-inflammatory cytokines, includes interferon gamma, interleukin 1 beta, interleukin 2, interleukin 6, and human necrosis factor alpha through PI3K, AKT, and MAPK cells in pathways. And next, this is another set of data from on the protein array analysis. Yeah. So what we can also see that this result also shows that we are able to suppress the immunotactic chemokines such as CCL5 and CCL21 while upregulating the expression of interleukin 4 and interleukin 10, which is anti-inflammatory cytokines, through PI3K and MAPK signaling pathway compared to healthier student group. Yeah. So this is the overall summary of the work that I've done in and in conclusion, I would like to say that the current study postulated the anti-inflammatory activity of EET against healthier cells, and therefore EET may be considered for the therapeutic purpose as potential treatment for inflammatory and neurodegenerative disorder. So for this data on this study has been published in this journal Content. Vivo studies on their memory neutropic effect of EET, and also to study the inflammation in the hippocampal region of the rats. Yeah. This paper is published in Life Sciences. You can have a look as well. And that's all from me now today. Thank you. And if you have any questions, please do email me, and because I'm ready so much.
Yes, sir. Thank you so much, Dr. Satishwaran N. Andy, sir, for your session on in vitro study on anti-neuroinflammatory properties of deoxyleftonin from Elephantophus scabrel. Hope most of the information what you have drawn in your research will be exploited to us. Most of the content is very good work, whatever you have presented to us. And thank you so much for your presentation, as well as thank you so much for the time what you have been with us. And coming to the next session, so I request Dr. Kali Rajan sir to be here online.
Yes, sir.
Yes, thank you, sir. Dr. Kali Rajan sir is Assistant Professor of JSS College of Pharmacy. He is a consultant, Constituent College, JSS Academy of Higher Education and Research, Deemed University, Mysore, Tamil Nadu, India. And I welcome Dr. Kali Rajan sir to take his presentation on COVID-19 activity of phytochemical constituents to the pepper, ginger, and garlic. It is an in silico approach. And I welcome sir to take over this session. Thank you so much.
So good afternoon. Is it audible?
Yes, it is audible, sir. You can proceed.
So visible also.
Yes, sir.
So good afternoon to one and all present here. So from the beginning of this current year 2020, we are all heard every time in all medias, social medias and newspaper the word coronavirus, because coronavirus will affect total world and all the world should be struggling by coronavirus. So as of now, there are more than 64 lakhs people affected by COVID-19. In India, more than 2.25 lakhs people are affected. So out of this, more than 3.8 lakhs people are died in the world. So in this pandemic situation, it is very difficult to discover new drugs for COVID-19 with all clinical trials, determination of all the side effects and adverse effect. That's very difficult. Since there are no any drugs or vaccines available for COVID-19, it is important to treat with some natural remedies without any side effects and which is used regularly in our diet. So we are determined the activity of phytochemical constituents of some natural remedies, which is already used for various biological activities by in silico studies using Schrödinger software. So let us go to our presentation. So our research topic is COVID-19 activity of phytochemical constituent of pepper, ginger, garlic and in silico approach. Because pepper, ginger, garlic we are used regularly in our diet, so we have to check the activity against COVID-19. So first introduction about this topic. So coronavirus is a life-threatening disease. It is a viral disease which is mainly caused by SARS-CoV-2, that is severe acute respiratory syndrome coronavirus 2, and MERS-CoV, that is Middle East respiratory syndrome coronavirus. So as on today, already I told us more than 64 lakhs people are affected in the world, and out of these 3.8 lakhs people have died. So till date there is no any confirmed treatment or vaccine for the prevention of the strategy against coronavirus disease. So that is COVID-19. So in this pandemic situation of COVID-19, it is very difficult to discover novel drugs with all clinical trials and determine all the side effects and adverse effects, etc. is very difficult. So due to this urgency of this situation, the drug repurposing, so that is already used drug we can use. It is widely accepted by fastest way to find out the novel drugs for coronavirus. So we have to identify some potential drugs by in silico drug screening experiment. So since most of the allopathic drugs have some side effect or adverse effect which is available in the market, so it is important to identify some novel or some natural remedies which is using regularly in our diet like black pepper, ginger, garlic. So we have to check the COVID-19 activity of these phytochemical constituents available in these natural remedies. So we have to identify some potential inhibitors for the following targets, which is used for COVID-19, that is SARS-CoV-2 main protease enzyme and RNA-dependent RNA polymerase, and the third target is ACE2 spike complex of coronavirus. So out of these targets, the main protease is the one of the important target which is responsible for cleavage of viral polypeptide into functional units, and also RNA-dependent RNA polymerase, which is critical for viral proliferation. So this is the structure of coronavirus. So it contains one crown-like structure that is called as spike proteins. And inside that in the wall there is main protein, the same protein, and inside that is RNA protein is available. This is the structure of coronavirus. So in our current research of finding active drugs against COVID-19 by in silico studies, so we have to check the binding affinity of various phytoconstituents of black pepper, ginger, and garlic against SARS-CoV-2 main protease enzyme. That is the PDB ID is 5R82, which is downloaded from the protein data bank by docking studies. So the chemical constituents of black pepper, that is the botanical name is Piper nigrum, and ginger, botanical name Gingiva officinal, and garlic, allium sativum. This already reported for many biological activities such as anti-diabetic, anti-tumor, antimicrobial, antioxidant, anti-inflammatory, and lipid-lowering effects. So, after based on this, lot of effect against many disease for these components of these natural remedies. So, based on the multiple biological activities of the above remedies, so we have to check the activity against COVID-19 by using computational studies. So what is docking? So docking is so already, so everyone should know the docking is a binding affinity between the receptor and ligands. So receptor, first we have to identify the receptor. So already I told there are three main target. Receptor is a target molecule. So that is for COVID-19 there are three main targets.
Sir, excuse me, keep your mic closer to you, sir. Keep your mic. Sound is very low. Thank you.
Okay, sir. Okay. Now it's okay, sir. Now it's okay.
Okay. Thank you, sir. Continue.
Okay. So there are first we have to select the receptor. The receptor is a target molecule. So the receptor we have to select the main protease enzyme. So SARS-CoV main protease enzyme. Then we have to select the ligands. So ligands we are selected many phytochemical constituents of pepper, garlic, and ginger. So these are selected as ligands. So this is a general mechanism. It is like a lock and key model. The general mechanism. So we have to select one receptor or target, and we have to select some ligands. So both are bind together by docking studies. So in this, so which is the best binding? So this is the binding pocket. The white color is a binding pocket. So which is sort of this ligand which is best to fit with this binding pocket we have to select, and we have to reorient by different conditions to best fit of that particular target with the ligand. So for your better understanding, we can play this video to understand.
Sir, can you please unmute yourself, sir? Can you please unmute?
Yes, yes.
Thank you, sir. Continue.
So first, this is the protein and ligands. So ligand first find the binding pocket, the active site of that particular ligand with protein. So mainly the ligand should be bind with protein with a different so this mainly the hydrogen bonding interaction is one of the important factor to bind the protein with the ligand. Then another one, hydrophobic pocket. So most of the compounds are lipophilic, so it is binds to the hydrophobic pocket. So the hydrogen bonding present in the hydrophobic pocket, so it is strengthen the binding or tightly binding the ligand with the protein. And all the aromatic part should be bind with pi cation. So pi stacking. So that is aromatic part should be between the aromatic part of the ligand with protein. So this is the understanding about this docking studies. So we have to use Schrodinger software for the. We have to use Schrodinger software for drug designing, in silico drug designing. There are various modules are involved in the in silico drug designing. So we are used Schrodinger Shoot 2019 fourth version. That is a master version. So it consists of many modules for doing the different activity like. So first we have to prepare the protein. So protein can be downloaded from the PDB protein data bank. And so we can use as such the protein. So we have to prepare the protein by using the protein preparation method. So the 3D crystal structure of SARS-CoV main protease protein we have to retrieved from protein data bank. The PDB ID is 5R82, which has the resolution of 1.3 Armstrong. Then the protein was prepared by using protein preparation method. That module is which is present in the Schrodinger Shoot. So Schrodinger shoot is a package. It consists of many modules. Then the ligands are prepared by using lick-prep model of Schrodinger shoot. And we have to do flexible docking, which is performed by extra precision mode, that is XP model, which is available in the glide. Glide model is mainly used for docking studies in the Schrodinger shoot. Then after the docking is over, we have to visualize by using the XP visualizer module, which is available in the Schrodinger shoot, to find out the post-dock analysis. And finally, so we have to check the in silico admit properties, that is absorption, distribution, metabolism, excretion, and toxicity properties of that particular ligands can be determined by quick prop model of Schrodinger shoot. And finally, the MMGBA assay, we have to find the binding free energy. So whether the stocking is more stable or not, that can be determined by the prime model MMGBA model of Schrodinger shoot. MMGBA is a molecular mechanics generalized bond surface area. So first we go for the protein preparation. So protein can be downloaded from the PDB file in protein data bank, that is RSCB website or NCBA website. The 3D crystal structure of protein, which is always contains with one co-crystallized ligand. Here the ligand is present is 6 ethyl amino pyridine, 3 carbonitrate. This is PDB ID is 5R82 that contains resolution of 1.31 Armstrong. So that can be retrieved from the protein data bank in RSCB website. Then the protein was prepared by using protein preparation method of EPIC module, which is available in the Schrodinger shoot. So we can use a protein which is downloaded as PDB file. We can use assets. So we have to prepare the protein. So in the initial stage, we have to delete all the unwanted water molecules, which is beyond 5 Armstrong, can be deleted. Then refine the bond orders and the addition of hydrogens. So any missing chain, the downloaded protein have any missing chain can be filled by using prime model of Schrodinger shoot. Then we have to minimize the protein by using OPLS3 force field, that is optimized potential for liquid simulation. So we have to reduce the energy by minimizing using OPLS3 force field with the RMSD value, that is a root mean square deviation value is less than 30 Armstrong. So then we have to generate the grid. So grid generation is we have to find out the active site. So this is the grid generation. So the active site, this box, the grid box is active site or binding pocket of the protein. So which is already contains co-crystallizer ligand. That ligand should be replaced with other ligands. Then we have to check whether the protein is good or not by using Ramachandran plot. So all the amino acids, all the dots represent the amino acids. It contains more than 300 amino acids in this protein. This is a monomer. So this is a single chain containing protein. So all the amino acids lies between this red and yellow area. So that is a good protein. So only 1 or 2 amino acids should be in the white area. So it is a good protein. Then we have to select the ligands. So ligands are selected as a phytoconstituents from the black pepper. So there are 6 ligands are selected like piperin, piperanine, piperetin, and piperlongumine, piperetin, and piper.in. Then the structure of major active constituents of ginger. It is from ginger officinal. So there are 8 ligands are selected. That is sorbol, 6 gingerol, 8 gingerol, 10 gingerol, paradiol, gingerol, and gingiberol, and phellandrol. So these 8 from this ginger we have to select. Then 8 ligands are selected from garlic. That is allyl alleine, allicin, ageone, vinyl dithiene, allyl methyl sulfide, diallyl sulfide, diallyl disulfide, diallyl trisulfide. So these are the 8 plus 8 plus 6. There are total 24 ligands are prepared by using lick prop module of Schrodinger shoot. So we have to draw the 2D structure, then it is converted into 3D structure by including all the stereochemical variations. Then the ionization and tautomeric state were generated between pH value of 6.82 shown by using the EPIC module of Schrodinger shoot. So on the final stage, we have to minimize the energy of all the ligands by using OPLS3 force field. Then the best ligand, the low conformation energy containing ligands are subjected to docking with the particular protein. So next is how to do the docking studies. So first we have to select the prepare the protein and prepare the ligand. Pepper from there are 6 ligands from pepper, 8 ligands from ginger, and 8 ligands from garlic that docked into the catalytic pocket of SARS-CoV main protease. That is PDB-ID already I told 5 or 8-2 by using glide model of sodium shoot. Then the best docked compounds can be identified by using the glide scoring. So the scoring function G score we have to obtain. If we get more G score means in the minus value we get more G score, that is good protein, good active compound. So we have to perform the docking studies by using extra precision mode of sodium shoot. So the algorithm recognized. So there are more favorable parameters like hydrophobic character, hydrogen bonding interactions, and the negative parameters like some steric interaction and other steric clashes should be involved. The XP glide score of active compounds were summarized in the following table. So currently there is no any standard drug is available. So we have to use hydroxychloroquine. That is temporary recommendation from many countries for the treatment of COVID. So we have to compare with this. So we'll see. This is the docking results for all the 24 compounds. So out of this, so when compared with hydroxychloroquine, that is G score is minus 5.47. So compared to this, there are 4 compounds are more active when compared to hydroxychloroquine. Like that compound piperidone, 8 gingerol, 10 gingerol, and piperonate. So these 4 compounds have good active than the hydroxychloroquine. So this is mainly due to this lipophilic evidence. So the first compound, the major contribution due to lipophilic evidence. The second compound, 8 gingerol, the major contribution due to hydrogen bonding and electrostatic force. So the first lipophilic endurance, hydrogen bonding, electrophilic force, and low molecular weight. These are the positive parameters to increase the activity. And some negative parameters are rotational penalties and XP penalties for negative parameters. So these 2 compounds, the Z1 is Z2, the SAG1 and 6 gingerol, the activity is reduced mainly due to the rotational penalty that we discussed in the next compound. So this is the best docking pose of all the compounds in the binding pocket of SARS-CoV-2 protease, main protease. So almost all the compounds are packed into a particular region. So this is the active binding site of that particular protein. So the docking, from the docking results, the highest active compound, the piperidone. So it's mainly exhibited interaction with COVID-19 main protease enzyme. The following residues like threonine 24, threonine 25, leucine 27, and histamine 41, serine 46, and these are the main amino acids are interacted with the particular ligands. So it is mainly interacted by using the hydrogen bonding and hydrophobic interactions. So the residue in the range of threonine 24 to glycine 189. So this is the range for the binding pocket. So 2D interaction diagram. So what are the amino acids are interacted with that particular ligand that can be shown in the next figure, and the hydrogen bonding interaction and other negative interactions are shown in the figure. This is the first top active compound. This is piperidone with COVID-19. So these are the active compounds, which is included. So this negative charge amino acids are in red color, the positive charge in blue color. So this pink color arrow represent the hydrogen bonding. So mainly the oxygen should be formed with hydrogen bonding with glycine 143. And this is pi stacking. This histamine 41 are interacted by pi stacking by using this aromatic ring. So this is the next highest active compound, 8-gingerol with the COVID-19 protease. The glide score is minus 5.88. This also best active compound. So this also affected the same binding pocket. So this is the hydrogen bonding interaction. So the compound, the second compound, 8-gingerol, that is second active compound. That is mainly due to the high hydrogen bonding interaction. The pink color dot represents the hydrogen bonding. So mainly this oxygen should be bonded hydrogen with the glycine 143. So this is the 6.19 Armstrong. This is a bond length for hydrogen bonding, and it also produces hydrogen bonding with the other water molecules. So some negative parameter already I told, the 6 goal that activity should be reduced due to this negative parameter. So mainly this dotted. So this is an active compound. This green color is a compound that ligand goal. In this all the single bond should be easily rotating. So that is a rotatable bond. So this is the mainly decrease the activity. So this is a negative parameter by rotational. So this bond should be rotated to reduce the activity. So this is the next. We will go for the ADMET screening. So ADMET screening. So absorption, distribution, metabolism, excretion, and toxicity of all the ligands. So we'll see all the. So mainly we have to check the molecular weight, dipole moment, donor. Hydrogen bonding donor, hydrogen bonding acceptor, and the QP log value in oil water system, number of metabolites, violations in rule of Lipinski's rule of and the percentage oral absorption can be determined in this table. So the recommended value. So all the compounds should be fall within the recommended values. The molecular weight between 130 to 725 is a good compound. So it's all the compounds are like this. Next, dipole moment within 1 to 12.5, and the number of hydrogen bond donor 0 to 6, number of hydrogen bond acceptors 2 to 20, and the QP log value in oil water system is -2 to 6.5, and the number of maximum metabolites 1 to 8, and number of violations of Lipinski's rule of is maximum 4, and more than 80% oral absorption is good, and less than 20 is bad. So we'll see all the values. So all are fall within the recommended values. So it is a good compound. So it is this is represents the safety profile of that ligands. So next, finally, we have to check the stability of the docking studies. So the docking binding energy should be more stable or not. So this is based on the value obtained from prime MMGPSA approach. So the binding energy should be computed by using this MMGPSA model. So already told that is molecular mechanics generalized bond surface area, which is available in the prime model of Schrodinger shoot, with incorporates the OPLS3 force field. So the energy minimization and VSC HGB solvent models to search the good algorithms. The accuracy of docking is confirmed by examining the lowest energy process in scoring function. So the lowest energy means that is more stable. So the glide score and MMGPSA free energy values are obtained in the docking should be increase the stability of if it is the lowest energy, it will increase the stability of docking. So it is tightly bind. So mainly these values is consist of DG bind value and Coulomb energy value, van der Waals energy, and lipophilic energy and hydrogen bond energies. So these are the positively contributing towards the binding energy in the MMGBSA. So this is the results obtained in the MMGBSA assay. So all the get negative. This is if more negative value should be the lowest energy, that is a more stable. So we will see all the maximum compounds. So you can obtain in TG bind and Coulomb energy, covalent energy, hydrogen bond energy is the negative value. That should be more stable docking. So the more binding affinity with the ligands with the receptor. And finally, summary and conclusion. So from the result of this in silico study, it exhibits the maximum chemical constituents. So mainly the 4 chemical constituents from black pepper and ginger. It is more active with the COVID-19 protein, that is main protease, when compared to the standard hydroxychloroquine. So the chemical constituents, so mainly 8 gingerol, 10 gingerol from ginger, and piper.in and piperazine from black pepper. These 4 active constituents are more active against COVID-19. So we can further refine this compound. We can get the good compound. So in conclusion, so we are consuming. This is we are regularly used in our diet. So we are consuming regularly in this situation by using of black pepper, ginger. So compared to garlic, these 2 have more active. So pepper and ginger we are used in the regular diet will increase our immunity as well as the prevention of coronavirus activity. So I thank all the organizers at the Department of Pharmacology of Chalabari Institute of Pharmaceutical Science, Andhra, and all other participants for your patience. So I thank all for giving this opportunity.
Thank you so much, Dr. Kalirajan sir. And you have given a wonderful session on the phytochemical constituents as well as the different software as well as all the things what you have shown. It is a very good session and informative. And I thank all the 4 speakers for their time today. And I thank Dr. Aditya Arya, who is an associate professor from Pharmacology and Therapeutic School of Medicine, Faculty of Health and Medical Sciences, Taylor's University, Malaysia, as well as Dr. Satishwaran and Andy sir, Senior Research and Development Associate, INQ from Malaysia. And sorry for the technical problem, we can't able to hear his audio to the maximum extent while his session is going on. And definitely we are sure we'll commence another session with Dr. Satishwaran and Andy from Malaysia, who is an Senior Research and Development Associate. And coming to the third session, I mean, Dr. Kalirajan sir, who is Assistant Professor from JSS College of Pharmacy, Mysore, as well as, and finally, Dr. Vijay Pandey sir, who is a professor from Chalapati Institute of Pharmaceutical Sciences, Guntur, Andhra Pradesh, India. And I thank all the participants. Without you, it is not possible to commence all these sessions. And a good response from you people also, a total of 846 participants too. And last day, it is rises up to 900 participants. Finally, we got 920 participants. And last day from 846 to 900, the score was increased a lot. And we also got participants from the side of North America as well as from Europe countries. So making this session as an international webinar truly and really. And I thank all the participants for being in this session. And mainly there is because for the support given by the college management, Sri Y.V. Anjaneyulu garu, as well as our free hand given by our principal, Dr. Ramarao Nadandla garu of Chalapati Institute of Pharmaceutical Sciences. Without their support, we can't go further ahead and we can't coordinate with all of the speakers as well as all of the participants. And regarding the feedback also, definitely you will get a feedback link to your registered email IDs. I mean, I think you have already registered by using one of the registration form that was already coordinated and sent with you. So definitely, I'm sure that you will get your feedback form links to your registered mails. And also most of the people who are not able to participate in this Zoom session, who have already got YouTube link, and most of you people have participated and view the session completely by using the YouTube link also. And thank you so much. I thank all of the participants and my colleagues, as well as the speakers, as well as our college principal and management body for making us to this much extent possible to organize this international webinar on recent advances in preclinical research. I thank you so much once again. Thank you all. Have a good day. Stay home, stay safe. Thank you so much.
Any questions? Questions from audience?
If you have any questions regarding these things, these sessions, some of the speakers are ready and you can ask questions by unmuting yourself. Thank you.